Welcome to The Explainer. Today, we're diving into a scientific revolution that's literally building human brain circuits in a dish, all to solve one of medicine's greatest and most personal mysteries. Okay, let's start with this number. Nearly 3%. That's about how prevalent autism is in the general population. And it's a figure that has shot up dramatically in recent decades, making this one of the most urgent puzzles in modern medicine. You know, for years, this has been the central challenge. Scientists could see the effects of autism on behavior, but what was actually happening inside the brain? That has been a complete black box. And that disconnect, well, it's been a huge roadblock, slowing down the search for any kind of effective treatment. So let's start with that central puzzle. How exactly has our understanding of this really complex condition evolved over time? Well, the thinking has done a complete 180. I mean, back in the 1950s, the refrigerator mother hypothesis actually blamed emotionally cold parents, which was just wrong. But today, thanks to things like twin studies and modern genetics, we know that autism has a very, very strong genetic basis, with hundreds of genes now linked to it. And this quote from Dr. Shinya Yamanaka, it just nails the concept. He says to think of it like a fever back in the 19th century. A fever wasn't one single disease, right? It could have been a virus or bacteria or even cancer. In the same way, autism isn't one single thing. It's a spectrum of behaviors that can spring from hundreds of different genetic roots. And that incredible diversity of causes, that points directly to the biggest roadblock we've ever faced in trying to develop treatments. And here it is, the fundamental problem. We simply can't study a living brain the way we can study other organs. I mean, think about it. You can't just take a biopsy from a person's brain to see what's going wrong at a cellular level, especially not during those absolutely critical moments of development before a baby is even born. This chart just lays it out crystal clear. Progress in medicine is so often tied to how accessible an organ is. We've made these huge leaps with things like blood cancers because we can easily get samples. But the brain, it has remained a black box. And that has dramatically slowed down our ability to find real cures for brain disorders. But what if scientists could finally get a key to unlock that black box? Well, a revolutionary breakthrough in stem cell biology did exactly that, and it completely changed the game. It all really started with learning how to keep embryonic stem cells alive in a lab. But the true revolution, the moment that changed everything, came around 2006 from a scientist named Shinya Yamanaka. He figured out how to create these incredibly powerful cells from any person with no embryo required. Okay, so the process he discovered is kind of like cellular time travel. It's amazing. You can take a mature cell, say from a simple skin biopsy, and you add just 4 genetic ingredients. These are the famous Yamanaka factors, and that cell rewinds itself back into a pluripotent stem cell. That means it can become any cell in the body, including, you guessed it, a brain cell that you can study.
So, with this incredible new tool, scientists didn't just stop at making individual cells. Nope. They started to build the fundamental components of the brain itself, right there in a dish.
First up were organoids. Now, these aren't just flat layers of cells. We're talking tiny 3D balls of tissue that actually self-organize to mimic parts of the human brain. And what's truly stunning, they follow their own internal developmental clock. If you keep them in a dish, they'll mature over 9 months, just like a brain developing in the womb. But Dr. Shinya Yamanaka's lab then took this a giant leap further, creating something they call assemblids. What they did was start fusing different kinds of brain organoids together to build working neural circuits. It's almost like building with biological Legos. So, this led the team to ask a really basic but profound question. Could they use this technique to build a circuit that performs a real biological function, like, say, triggering a muscle to move? To do this, they needed to assemble 3 distinct parts. You've got an organoid for the cortex, that's the command center. Then you have one for the spinal cord, which is the relay station. And finally, you have a piece of human muscle to act as the output. And it worked. Incredibly, it actually worked. The cells organized themselves, they formed the right connections all on their own. So when the researchers stimulated that cortex organoid with a bit of light, the muscle at the other end of the circuit twitched. They had built a working biological pathway from scratch. Now, this technology isn't just some fascinating lab experiment. It is now a powerful, powerful platform for understanding devastating genetic diseases and developing real-world cures. Take Timothy syndrome, for instance, a very severe genetic form of autism. By taking skin cells from patients and building these little circuits, scientists were able to pinpoint the exact biological problem. A calcium channel in their neurons was staying open just a little too long. And because they could see the problem, they could develop and test a therapy that reverses that defect right there in the dish. And that therapy? It's now on its way to the first ever clinical trial developed entirely using this method. This approach is completely transformative. I mean, it takes what we used to see as these mysterious, purely behavioral disorders, and it reframes them as concrete biological problems. Problems that we can actually see, that we can measure, and that maybe we can actually fix. Which leaves us with a truly provocative thought. If we can rebuild these intricate, broken brain circuits in a dish, what's the limit to what we can understand and ultimately fix in ourselves? This revolution is really just beginning, and it's opening up a whole new horizon for conditions we once thought were completely unreachable.